we're just going to see, well, a different tool outside of R, just using Linux, to pretty much perform the same thing you did with David yesterday. How to do a multiple sequence alignment, yeah, from the Linux command line, using pretty much the same algorithm David used. Cluster. Yep. So if you could please open your BioLinux boxes, mount yesterday's MSA directory, where you had the multiple protein. So are we all there? We'll start earlier than usual because we usually don't have enough time in a day to finish all the things we need to do. So nothing has been done yet. Just turn on your VirtualBox and mount your MSA directory from yesterday's work with David. You mount it. If you don't remember the command to mount it, just ask us. You should know it by heart by now. So for everybody who doesn't remember, we are importing from Windows into our virtual machine the MSA directory we used yesterday for multiple sequence alignment. The first example today is going to be to repeat what we did with yesterday with David, but instead of using R, using a command line tool. A command line tool that you already have installed in your machines, in your virtual machines. So the first thing is create a directory where you are going to mount the Windows directory. So basically create a destination directory. Remember, you had your day 6 practicals yesterday. Inside of day 6 practical, you had MSA, more R, and there was another one. Yeah? And what we're going to work with is the MSA directory. But that's on Windows. You want to be able to access that from inside your virtual machine. If you try to look, you won't find it. Why? Because you need to share it with Linux. Right? How do you share it? You have to mount it. We've been using the command mkdir to create a directory. For example, under Documents, INEA 08. That's an example, yes? El de MSA. Ahora, usearch. Recuerda que tenías nada más la carpeta de usearch. Ahora lo que vamos a hacer es la carpeta de MSA. Es una nueva. Sí, no, el user no necesitas montarlo, solamente el MSA ahorita. Tienes que crearlo como dispositivo compartido. ¿Sí? El MSA, ya ahora tienes que crear la carpeta MSA. Remember, guys, to use tab as much as possible so that typos are not made, or at least are reduced as much as possible. Yes? Oh, that is cool. Did you? Wow. Let's go in YouTube. We enjoy it. I have never seen that. What are you doing to your computer? What are you doing to your computer? I had never seen that under virtual machine. ¿De cuál? ¿Del virtual machine? Ah. No, yo también lo actualicé anoche, pero no me salió eso. No, pero no lo dejaste actualizar, o sea, lo puse para lo retrasé. Está raro, pero no importa, igual está funcionando, tranquila. Esa pantalla de inicio donde tienes opciones, cuando tú instalas dos sistemas operativos, por ejemplo Windows y Linux al mismo tiempo, ese es lo primero que te va a salir cada vez que enciendas tu máquina. Es decir, ¿qué quieres? ¿O usar Windows o usar Linux? Y ya te escoges. ¿Sí? ¿Cómo dirías? Ajá. Haces lo mismo, el mismo comando, chmod 750. 750 significa todos los privilegios para mí, ejecución y lectura para los de mi grupo y ningún permiso para externos. ¿A qué? A todo lo que termine en .r. Sí, el asterisco es un wildcard, significa que empareje lo que sea, o sea que haga match con todo, ya, pero que todos los archivos tienen que tener con .r. Los que estoy seleccionando tienen que tener con .r, sí. Paciencia, paciencia, paciencia. La paciencia es una virtud. Patience is a virtue, guys. VBox SF. VBox, here the minus T, VBox SF. 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 Si te falta la F. No, ahí no. VirtualBox SF. Ah, no. S, ya tienes la S, solo te falta la F minúscula. No, después de la S. SF, ahí está. Perfecto, borras esa F mayúscula de ahí, no la necesitas. También borra esa F de ahí, porque no es ningún directorio. Y acá no te olvides de poner el directorio de destinación, ya, es el que acabas de crear. Y fíjate dónde lo has creado. Documents, que está bajo Manager, que está bajo Home. Es Home, Manager, Documents, MSA. Alright guys, I know it takes a little bit of time. For those of you who have already reached this point, yeah, I encourage you to just type the word CLUS, C-L-U-S, and double tap. Just double tap. Before it actually selects anything, you will see that you will be given a set of options. Yes? No such device. Did you hear? So, you've seen that when you type class and you just tab it, you'll offer with a series of options, right? Do any of you remember what was the algorithm you used yesterday in R to do the multiple sequence alignment? Oh, okay. All right. But I think you mentioned it. Well, I guess you remember you used the default, which is the most common multiple sequence alignment algorithm that there is around. Probably most of you have used it, even though you're not sure. Is this mine? Oh, it's yours. Ah, there's mine. Is this mine? Yeah. Oh, that's mine? Yeah. Where did I leave my bottle? What did I do with my bottle? Oh. So this algorithm is called CLUSTAL. If you've tried once or twice, if you go on the internet, you Google CLUSTAL, yeah, you will see that you have a very common page, right, where you can just run multiple sequence alignment online and get your alignments in whichever format you want, because there are many formats to represent an alignment, yeah, and then you can use that alignment for several things. Now, there are some caveats to this, there are some limitations to this. First, there are kinds of alignment. Yeah, you have global alignment and local alignment. Global alignment is when you're trying to compare or to align sequences that are pretty much the same length. Why? Because global alignment will try to force all of the sequences involved, yeah, to be evenly matched from start to end. Local alignment, on the other hand, it's ideal when you want to align a very short sequence against a larger sequence. Why? Because it will find the optimal, most consecutive region where both sequences align, regardless of their length. It will not try to push the alignment or to force the alignment into be evenly spread across the longest sequence. This is important. Why? Because we can introduce bias in an alignment when we push for a global alignment approach if we're using sequences that are very, very different in size. If one, yes? I'm sorry? Global alignment, yeah, is when you want to find basically you're telling the algorithm, hey, I am assuming these two sequences are very much alike, so they should be aligned from the beginning to the end. So the algorithm is trying to force the alignment to go from the beginning of those sequences to the end of those sequences. If one sequence is longer than the other one, then the shorter one will have matches spread across the entire longer sequence, yeah, instead of finding a local maximum. It will find the best score, yeah, but trying to optimize the length, trying to use the entire length of the longer sequence. That's why you'll get weird alignments. We could make a test, but it's not gonna be the very quick. So, but if you have sequences that are significantly different in size, like one is 3 or 4 times longer than the other one, than the shorter one, you should go for what we call local alignment. Local alignment is optimized to find the most localized alignment that produces the highest score. Needless to say that when you want to build, or when you want to find relationships, like evolutionary relationships or just sequence identity, determine sequence identity between two sequences, the most ideal scenario is to use sequences that either belong to the same orthologous, the same family, the same orthologous genes in different species, yeah, so that the average length of all the sequences that you're aligning is pretty much the same. It may vary by a few nucleotides or by a few amino acids. Yeah, one might be 300, the other one might be 340, the other one might be 290, but they are all very close to each other. That's what we use this multiple sequence alignment for. For these short sequences, well, they don't need to be short, but at least they need to be the same length or close to the same length. And why? Because we're trying to see the multiple sequence alignment will give us some clues as to how this relationship, the sequence of this the relationship of these sequences has evolved, has changed, how similar they are, and maybe even help us reconstruct the evolutionary history of these proteins. If you've ever seen a tree, either a phylogenetic tree, a cladistic tree, you've seen that it is based on sequence similarity. Whether it is DNA similarity or protein similarity, but it's based on the similarity, these similarity matrices. And those similarity matrices are calculated based on a primary alignment or a multiple sequence alignment. So the program we use, or the algorithm, the most common algorithm for doing this multiple sequence alignment, as David mentioned yesterday, it's Clustal. And we have different flavors of Clustal. You'll see that you have Clustal X, Clustal W, Clustal Omega. The most modern version is Clustal Omega, which has optimized subroutines to make the alignment not only more accurate, more sensitive, but also faster. Clustal X is the previous version, and Clustal W, I don't think I ever used it. We will be using Clustal O, which stands for Clustal Omega. It's the latest version of the Clustal algorithm family. So this is what I was asking you to do. Clustal O minus help, and you'll see that there is a big help. It will tell you how to use this tool, what it requires. Now, Clustal O is a program, okay? It's a program, it's optimized. You don't have to install it because it already comes as a default on BioLinux, but in case you start using a different machine, you'll probably have to install it. The easiest way to install any program is to go to the page where it's meant to be and look for the keyword, binaries for Linux. If you're going to use a Linux machine, go for the one that says binaries for Linux, because that means that the programs are already prepackaged to run natively from wherever you copy them. If you copy them to my documents, You just go into the directory where you copied it, and you just put /, write the name of the tool that just downloaded, yeah, and -- help, and it should work. Most cases, it will work straight away. If it doesn't work, you don't have to do it right now, I'm just telling you. If it doesn't work, then you probably have to install it from scratch. That's a bit more complicated, but it's nothing out of this world. All right? We're not going to touch how to install programs in Linux, because that's out of the scope of this course. Yeah, but I just wanted you to know that it's not very difficult. So in this case, you already have a prepackaged or precompiled program, which is called cluster-low. And cluster-low, what's going to do? It's going to do multiple sequence alignment, just like you did on R yesterday. And now, if I remember correctly, you had two lists of genes. You had trypsin and chymotrypsin that you downloaded from Swissprot. Yes? Right? Do you remember, do you still have those files? Please check that the directory that you have mounted, you still have those files. Trypsin.fasta and chymotrypsin.fasta. If you don't, please copy it over from your neighbor, from the people right next to you. Remember that those FASTA files should be inside of the MSA directory, not anywhere else. You got them? Excellent. So that's what we're going to use as input. Those sequences are not aligned. It's just a collection of sequences. What we want to get is the proper alignment of those sequences. So that's what we're going to use cluster-low for. So there are a lot of options in cluster-low. We're going to just be going over the basic ones, the ones that you're going to need for pretty much everyday work. A typical invocation would be cluster-low, my I, which is my in6.fasta. Can somebody guess what this is? What this file means? The input. And what would be your input? Chymotrypsin.fasta and trypsin.fasta. My O, it's my outsex.fa. What are we going to get there? The name is a bit misleading. The name is a bit misleading, but it's pretty much an alignment file. Why is it being called .fasta or .fa? Because there is an alignment format also called the FASTA alignment format. It looks pretty much the same as the FASTA, except that if there are gaps in the alignment, you'll see a dash in the middle of a sequence. Yeah, but and all the sequences are going to be the same length because they already have been aligned to the same length, to the longest one. Yeah, but keep in mind that even if it says .fa, the output is an alignment. We're going to change the format of the alignment. Now, most of the other inputs are not going to be discussed. For example, HMM in, it's HMM input files. If somebody is curious about what HMM is, yeah, those are hidden Markov models. Hidden Markov models are statistical tools that have been built based on previously made multiple sequence alignment that tells us basically what are the most likely paths for the alignment to be built. Yeah, that's pretty much the sum up. Yeah, these are just probabilities that you build into a file. You use this a lot when you're trying to find hidden motifs or conserved motifs in a protein or in a protein family. Yeah, but in this case, we're not going to use that. We're not going to work that way. So D-align, profile 1, profile 2, or is profile, it's basically different profiles to improve either the speed, yeah, or the accuracy of the alignment, because we're giving them presets. to the cluster algorithm to tell them, hey, use this path for the alignment. First, you wanna find this motif inside of it. Try to align that motif instead of finding new regions that are more likely to be aligned. We're not going to do that. We're not forcing the alignment to go in a specific pathway. You're just letting the alignment do its thing. Because right now we don't know anything about these sequences. That's our assumption. This is the first time we're seeing these sequences. For some reason they look alike in BLAST, and we've downloaded them, and we're trying to see how well they align to each other. Now, this one is important, -T, or --, sec type. Here you tell it what it is. Is it a protein? Is it DNA? Is it RNA? Why? Because it will use a slightly different algorithm tweaks in order to improve the efficiency of the alignment for each type of protein, RNA, or DNA molecules. Protein has an alphabet of 20 characters, 20 different amino acids. RNA has an alphabet of 4, sorry, where thymine is replaced by U. What was it? Yeah, uracil, yes, that's it. And DNA, you have TCGA. Now, -- in format, in FMT. Here you said what is the input you're doing. What format is the input you're giving to this file? And we all know that we're using FASTA format. We can just leave it as default because it will be auto-detected. And these tools are pretty good at auto-detecting what they're doing. Now, if we want to change something about the alignment algorithm or the clustering algorithm, we can try to use all of these. Personally, I've never used them. Right? But if you had previously built, for example, a guide tree to guide the alignment, it's also a suggestion you give to the aligner. You can, using a different algorithm, like muscle or something else, then you can use that previously built guided tree, or maybe you have now 1,000 sequence and you build a tentative tree using just 8. And you want to use that 8 sequence alignment, as a model to guide and to speed up the alignment of 1,000 sequences. And that's when you use guide tree. We're just using 9 sequences or 10 sequences, so it's not a big deal. PyLab, sequentially aligned sequences 1 to the previous alignment of the other 2. Use full distance matrix for guide tree calculation. So since we are not giving it a guided tree, it's going to calculate its own guided tree based on the data we're giving it. For during iteration. We can use several iterations as well, just to improve the accuracy and sensitivity of the alignment. These ones I don't think I've ever used them. Use Kimura distance correction for aligned sequences. I think Kimura distance correction is for specific sequences. I'm not sure what it is. And the alignment outputs. -O, the name of the file you're going to be writing, just like at the beginning. My alignment dot something. Usually the dot something, it's the format that you're using. Out format, and you'll see that you have a plethora of different alignment formats. The most common one is this one, CLU, which stands for cluster format. But you may also want to do Philip if somebody has already worked a little bit on phylogenetics or something like that. You've seen that Philip is a very common format for multiple sequence alignment and for trees. Stockholm, some tools do require the Stockholm, but this is going out of fashion. Vienna, I don't think I've ever used Vienna. And you also have the A2M, which is FASTA, which is similar to a FASTA file, except that whenever there is a gap introduced, you're going to see a dash. That's it. Remember that the guiding tree that you produce from multiple sequence alignment, it's not a phylogenetic tree. There is a big difference between the two of them. Yeah. This one is just a distance tree. That is based on the genetic distance, the number of mutations that have happened between the sequences you're comparing. You're generating a similarity matrix and using that to say which ones are closer to each other. A phylogenetic tree is something a lot more elaborated. Basically, you do iterations because from each similarity matrix, you can produce pretty much endless different trees. And you have to optimize the way you build a tree. You either use maximum parsimony, maximum likelihood. Yeah, exactly. And after doing that, you have to do it based on models. How do you expect a DNA sequence to evolve? And for example, there are things that are not necessarily so obvious unless you're into the field. The fact that, for example, transitions and transversions. Yeah, transitions are twice as likely as transversions to occur, even though the number of possible transversions is 3 times the number of possible transitions. And yet the transitions are way more common. Why? Because of the molecular biology or the biochemistry behind how transitions work. Yeah, so these are things that have been seen empirically from experiments. Yeah, and they have to be modeled into phylogenetic tree builders in order to make the right assumption. So just the fact that a guiding tree for alignment or a similarity matrix that produces a tree looks similar to a phylogenetic tree doesn't mean that they are both the same thing. Is that kind of clear? Yeah. So we select the output file, the output format, residue number. Basically, this tells us how many characters are going to be printed as a total alignment. We're just going to keep it as print everything. How many residues per line? That is how many residues we're going to see in a single line before it breaks and goes into the second line. Input order, if we have an order that we would like our sequences to appear, this is the place to state that. We don't. And iterations. How many iterations do we want the guided tree to be built before choosing the optimal guiding tree to build to cluster our sequences? Usually for a first approach, 1 iteration should be all right. If you're going to be very detailed, you can do 2 or 3. I wouldn't go past 3. Now, max number of sequences. Maximum number of sequences, number of sequences allowed. We don't want to put a limit to it. Besides, we're using very few. Maximum length of the sequences. Do we want to truncate our sequences to shorter sequences so that the alignment works faster? Or we don't. We probably don't. Miscellaneous. Auto. Set options automatically. Most of these have already defaults that work for most proteins, for most sequences. So we can use those defaults. But it's important that we understand what we're doing. Threads. We've already been through this. All tools have this. This is to speed up. The more threads we use, the faster the alignment is going to run. Yeah, but also the slower our computer is going to be because we're using more resources. Pseudo-parameters, no, we don't have any of these things. So let's go for it. So we're going to do close hello. Yes. Yep. I'm sorry, what distance? The other distances don't depend on the Kimura one. Yeah, basically, yeah, this is not an evolutionary model. This is not a devolutionary model. That's what I was telling. It's like there's a big difference between phylogenetic tree and just multiple sequence alignment. Yeah, the multiple sequence alignment builds a guiding tree just to speed up calculations. Speed up calculations and do the alignment as fast as possible. But if you want to do evolutionary models, it is a lot more complex than just that. All right? Now, Kimura, as I told you before, I've never used it before. I'm not entirely sure how it looks. Do you? Yep. But this is probably just a statistic or just a way to speed things up. If you have previously calculated Kimura distances from a set of sequences that are related, you may be able to use it. Yeah, but since you say the default is don't use it. Why? Because Crosstal doesn't need it. The algorithm doesn't need it to actually work. So the first thing was to set up, select my input file. So my input file first is going to be trypsin.fasta. Please tab so that you can autocomplete and there are no typos. -O, your output. Let's say we're going to see trypsin.clustal minus informat equal FASTA. Just making sure that's the right way. Informat, okay. Well, you don't need to write FASTA, just FA. My minus out format should be Clustal. There we go. Out format Clustal. Oh, minus. Okay, just be careful with this. Minus, and minus. And that should be it. Oh, minus threads. I'm going to use 4 because I like to use 4. Yeah, you think about the capabilities and the number of threads you already have on your computer. If you have 8, you can use 4. If you have less than 8, just use 2 or 1.
Sometimes we use 1 minus, right, to specify a parameter, but sometimes we use 2 minus. The difference is when you use a one-letter code. When you use a one-letter code, it uses 1 minus. If you use a word, like here minus log, it uses 2 minuses. That's the only difference. That's what Linux uses, not only in bio programs, in all kinds of other programs that you can find.
Now, just for the sake of experiment and to see what's going on under the hood, we're going to add this -v. Can you see it in the back? -v means verbose. Most bioinformatics programs and most programs in Linux also have it. What does it mean? It's going to give you a detailed description of what's happening while you're running your script. Right? It's going to be verbose. It's going to talk to you all the time. Right? In this case, you usually want to skip it because you already know what's going to happen, but in this case, we're going to run it with verbose so that you know what's going to happen. This is probably the first time you run it online. Oh, you run it from the command line. Or have you run it before from the command line?
Yesterday, we used it also when we used Run.
Oh, yeah.
But verbose 6 was false, so you wouldn't see it in R.
You see? This is what happens when you don't tap. There we go. Minus, sorry. Type. Yes, this is the one I was forgetting. The type. Sequence type. Protein. Sorry, that's my bad. Sorry? Yeah. Just used to it. Yeah, sorry about that. Minus T, it stands for type of molecule, and we're using a protein. Minus threads, that's the number of threads. They should use the same flag for everything. Yes? Oh, sorry. I think there's a free spot here if you'd like to come closer. Now, you do understand why I did the change, right? I had an error that was telling me the sequence type kind 4 doesn't exist. Why? Because I mistakenly put minus T, which was expecting something like protein, DNA, or RNA, and I gave it a 4. I just assumed it was a different parameter. Yeah? That's why I changed it. Yes? No, they don't have to be in this order. That's what the flags are for. The flags are telling the program, after you use this, minus O, the following argument is going to be the name of your output file. So you put the block, minus O, trips include, at the end, at the beginning, or whatever you want, it doesn't matter. As long as all of these flags come after the command. The command must come before that. You cannot put minus E trips in fasta clostalo, because the Linux is going to think that the first word you use, which was minus I, yeah, is the command. And it's going to tell you, hey, I cannot find a command named minus I. It doesn't exist. Because you have to put the command first, clostalo. There we go. It was very slow. So it's going to tell you everything that's going to happen. Have you guys run it? Don't be afraid. If there is an error, typo, anything, the program is going to detect it and it's going to tell you, hey, this went wrong, and you'll be able to read it and change it accordingly. I know it feels a little bit, using Linux, getting used to it, it feels a little bit like when we teach our parents how to use Windows, right? And we teach them, and they call us every time and say, hey, I'm having this error, what do I do? What does the error say that? Oh, it says that I should shut down the computer. Then what should you do that? I should shut down the computer? Yes. Yeah, it feels like that. But yes, trust your warning signals. Trust what the program itself tells you what to do or what not to do. But for that, we need to get used to reading what the warnings are, what the errors are, and trying to wrap our heads around it. Sometimes we're not going to understand a thing, because these are like compilation problems, etc., that we probably have no idea how to deal with. Yeah, but more often than not, for example, all these commands that is here, no command clostalo found. Why? First, it was a typo. Then I fixed that. I say, hey, fatal unknown sequence type T. Oh, there was a mistake in my command. I go where I use the 4 and I say minus T. What? Minus T doesn't expect a 4? Go back to the help and see, oh yeah, minus T doesn't use numbers. It uses a format, which is protein. And that's how I keep fixing my own errors. But I have to read my errors. So now it's done. In my case, just as I asked, using 4 threads, cool. Read 7 sequences, the type protein from the tripsin.fasta, not more than 7 sequences. Then cluster size 100 turn off. Calculating pairwise k tuple distances. This is part of the algorithm. Don't worry if you don't understand it. I don't understand it either. K tuple distance calculation progress done. So there is a distance calculation between the sequences that has been finished. In how many? In 1 microsecond, it looks like. Yeah. Guide 3 computation done, so it's already produced the guided 3. progressive alignment, progress done, CPU time, all this, alignment written to tripsin.clue. So the alignment has been written, and what tool should we use to read an alignment file or just any file, in any text file in Linux? You can just beam, sorry? That is the variables, yeah. If you were to skip the variables, you probably wouldn't have anything written. If you remove this, yeah. You see, it's telling me fatal error. Cowardly refusing to overwrite already existing file tripsin.clue. Why? Because the program is saying, no, this file already exists, I don't want to overwrite it. Yeah, and it's kind of, it's a funny way because these errors are written by the programmer. So the programmer finds a way of saying, oh no, I'm too coward to do this. So yeah, I find it funny. So if we were to use what they are telling us, force, please be aware that if you use force, you're going to overwrite whatever what it's telling us. Yeah, you see, they even did use minus force to force overwriting. There you go. With no minus v, there is no verbose output. So you don't know what's going on under the hood, but it just works. It is like a nice black box where you put something in and you get something out and you don't care about how it worked. So you'll see this is the alignment. Yes? Mm-hmm. Suspended means? What happened? Oh, yeah. It depends. How do you exit? It depends on how do you exit. If you exit like this, you shouldn't have any problem doing it again. But if you, yeah. Don't worry. What's happened is that you left this open. Just do ls minus la. Yeah, and you'll see that you have some files that start with a dot. Yeah, those are, in Linux, those are hidden files or temporary files. For example, in this case, you have these two. Tripsin cluster, swap and swap. Yeah, erase these two. Just remove these two. Remove dot tripsin whatever, and those two. Yes, erase it. Yes, erase it. You can remove it. Yes. Cannot remove a text file VC. Do you have any other? Have you opened it somewhere else? Sure? Doesn't look like it. That's weird. Usually you erase these two, and then you're able to look into that. That's when you had a weird exit of your bin. But yeah, I don't know why you can read it. If you want to do it differently, you just copy tripsin.postal to a different directory and just see it from there. Yes. Oh, just 1 second. Yes. La actualización. La actualización tienes que darle doble clic. Tienes que correr la de Windows. De Windows de donde la descargaste, doble clic y automáticamente va a encontrar tu instalación y la va a actualizar. Okay, you're all seeing this? Yes. Mm-hmm. You do. This is similar for. Yep.
Okay. But for example, you have too many options, but you don't know exactly which
Oh, you have to go online most times. You have to go online for all of that. Yeah. There is online, you will find a very thorough manual. No, it's the same. You're exactly where you were. The only thing I've done is just I just put the help.
Yeah. So, and my other question was, so if I stop it in the right
Minus V.
yes, minus V,
Yep.
I can go do that.
Yep. There you go. Finish. Take a look at the alignment. Yeah, the most likely scenario is you'll have to go all the way to the original Clostal paper. Yeah, the original Clostal paper is going to give you a thorough description of what each of the parameters is, and yeah, because this has a lot to do with how the algorithm works and how the statistics of the calculation are being met. It is a lot to go into. So yeah, as David was mentioning, this is exactly the same thing you did yesterday, just using a different tool. Oh, you'll see here. In the minus help, in the minus help, you can see what are the other formats that it receives. So, if you look, the alignment has been split in several blocks, right? That's because we didn't set the maximum or the minimum number of characters to be in a single line, yeah, and therefore it just chop it at I think the default is 60 characters is the default per line per block, yeah. So this is the alignment of the first 60 characters. Now, how do you know what are the conserved sites and which aren't? Yeah, it's by the consensus line here. Everything that is a star is exactly the same. Yeah, that means that all the alignments have the same residue in that position. If you don't have anything like this, it means that all the alignments are pretty different. What is the convention for dot and double dot? I think it has something to do with, yeah, the percentage. Sorry. Yep. Well, usually these alignments can be used for downstream analysis. This kind of sequence alignment can be used for phylogenetic inference. This can be used, yes, you can use the alignment for phylogenetic inference, but it's going to use a different algorithm. You just use the alignment as a reference. Say, okay, this is the way they're aligned. Now we can apply evolutionary models that we trust in order to model and select which is the most either parsimonious or the most likely evolutionary path for these proteins, which is.
All these terms in the lecture that I provided, the presentation that I provided in the lecture folder. Or should we get into the lecture?
Yeah. So the exercise right now is to do exactly the same thing, but for the chymotrypsin FASTA files.
This is the first exercise. The second exercise is to concatenate the chymotrypsin FASTA file with the trypsin FASTA file and then run another
Okay. Hey guys, in case you're wondering what the consensus line means, here it is. Ahora sí, ahora. Sí, a mí me parece que no debería haber problema, pero ¿por qué está haciendo este problema ahora?
So, we run this now on trypsin, right? Run multiple sequence alignment on trypsin. Second exercise is
Finish?
concatenate the
Finish?
FASTA files for trypsin and chymotrypsin, and on the concatenated file, running out multiple sequence alignment.
Let me know if it works. Hopefully. Cut. Sorry. That's a good way to go with how cut works. Cut.
Or you get a lot.
Yeah. Cut. File 1, file 2, and then redirect to a new file.
So if you write the command cat, the first file dot faster, the second file dot faster, it will print it out to your screen. But you want to save the file. So you write cat, first file, second file, greater than sign, and then a name of a loop file.
A file that doesn't exist. You can put it your own names .fasta. You can put it exercise 2 .fasta. Oh, this is the explanation of what the consensus line in the closed alignment means. Yes. Ya, muy bien. Ahora hace el otro ejercicio. I think you already have a good idea of what the colon, period, and asterisk mean in the consensus. So I can just move it away.
Pretty obvious.
I wasn't sure.
No, I mean, when you look at it, you see that you have an asterisk in the 100%.
Oh, yeah. The asterisk, yes, but the period and the dot, you have to actually look into the line and say, yeah, look at this. TAFLL. Oh, LLLIIL. So I think, yeah, it has something probably to do with the percentage, but exactly where is the threshold? I prefer to just look at.
Yeah, you're right.
Yes. Si quieres. Hacer permanente, sí. Sí. Ahí está, MSA. Ahora está. Acá te dice. ¿Qué pasa? Más de 50%. Ah, ya, no te preocupes por eso. Le has asignado, tiene 3 GB, le has asignado más de 1.5 GB a tu máquina virtual. Have you all copied the day, the INEA 08? Yes? 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 Okay, cool. Yes? Oh, yeah, don't worry. You're in Beam. You have to. Where are you? Oh, you put it cut, okay. Yeah, don't worry. What was your command? That's okay, don't worry. What it is doing is actually concatenating this, and whatever else you put it. For example, you put F, right? Minus F. It's all you F, no such file directory, because it's interpreting F as if it was the name of a directory. Or of a, yeah, you just want to concatenate this and let's use this. Oh. There you go. This greater than sign, yeah, it means this, instead of printing on the screen, save it into the next file. And here you write a file, the name of a file that you want. Exercise 2.fasta comes to mind. Yes? Yep. Output. Yep. Yeah, excellent. You probably want to Custal. It says Custal Omega. This is the one. You can do it here. You can go to Wikipedia. You can go to Biostar, different output from Custal Omega. Oh yeah, it's just that I look for it in different ways. I just, yeah, what I did is just writing in Google, understanding the consensus format in Custal. Understanding consensus in Custal. And then it's going to give me a biology stack exchange, and that's where all these things appear. How's it going? Aha. There you go. Yep. Looks good. Easy. This is a FASTA, isn't it? Okay. Have you aligned them? I've already checked. You cannot, it says the device doesn't exist. So it says, one thing failed with real particular. What I've seen this before, and the solution was to restart the machine. Yeah, we restarted, but we've restarted and we've reinstalled this twice already. So I'm not entirely sure what's going on.
You cannot even run MS.
If we go here and we go L, there's something wrong there. it make sense to forcibly remove documents? I think here started the machine from the OVA, so nothing of the pre-configuration should be there. Unless it leaves a trace in the window. That's going to be fun. Let's try something drastic. Sorry.
Yeah? What is the result?
Thank you.
What will
Now,
you see?
si supiera cómo resolverlo ahorita. Solo Joao. Joao tenía un problema distinto, pero relacionado. No puede montar la misma carpeta otra vez. No la puede montar por alguna razón. Pero él ya reinstaló todo de cero. Igualito no puede. Sí. ¿Por qué? No sé. ¿Qué marca es? A ver, voy a chequear si esto llega. ¿Puede ser? Esto es Shiva. Podría ser eso. Eso podría ser. Okay, guys, do you have any questions so far? Everything clear? So, as David was mentioning, yeah, there is a reason behind why some, what appears to be distance proteins have conserved sites. Does anybody want to guess, or from your own experience, want to share what a conserved site in a protein or different proteins mean? Why would disparate proteins, or just different proteins from different individuals, would have common regions? Regions that are highly conserved when you align them. Why would that be? You can share it from what you already know, from your own experience, from your classes, from whatever you already know. And David has already mentioned it, but I think we were all too distracted trying to make things work, and maybe we just missed it. Why would you say this happens? Higher up. Up. Increase your volume. Exactly. Or at least this small.
Hola.
So at least these small parts that are very conserved, they probably have an active role in what the protein function is. The most often, the most common scenario is that these belong to highly conserved functional domains. They serve a very specific function within the protein. And since this function can also be replicated in another protein, yeah, for example, let's imagine this scenario. We have a part of the sequence of a protein that is going to interact with DNA. All right? How many different proteins, protein families, will interact with the DNA? Several. And each of them is going to have a different reason to interact with DNA. Some of them are going to cut them. Some of them are going to modify them. Some of them are just going to promote transcription. Right? But they are all going to be attached to DNA. So you may argue, yeah, that there should be a specific sequence that could be highly conserved in all these proteins that have to have some kind of tight connection with DNA that makes this possible. And that could be a conserved domain, because that function or that subfunction should be part of all these proteins that have to interact directly with DNA. That's just an example that I can think of. Right? So yeah, the fact that they share similar subfunctions or even similar functions says that it's likely that they will share these functional operational motifs inside of their proteins. And when you align them, those motifs are the ones that are going to be highly conserved. The rest might be completely different, but these motifs are going to keep showing up.
So empirical evidence has shown usually that more commonly the recent changes, the new radical changes on the periphery of the protein. But when you look at the cellulose approach of the protein, think about the biology then. But just because something is at the beginning doesn't make it as peripheral. It can be actually the center, right? All the tertiary structure. You cannot know that by
We can guess it. We can guess it. For example, this is a very rough guess. It's like, would you expect that hydrophobic amino acids would be on the outside or the inside of a protein, of a folded protein? It is. Why? Because they're trying to escape from water, right? Yeah. But what if you're not in water? What if you're in the middle of a membrane? Where would you expect the amino acids, the hydrophobic amino acids to be? In the outside or in the inside? Close to the membrane, right? Yeah, so this is just a rough estimation. There are already several tools, online tools and also command line tools that let you predict with around 1 or 2% accuracy, right? Yeah, something around that. 1 or 2% accuracy of. The other thing is that a lot of people are working on, yeah, and they're trying to use like artificial intelligence and neural networks is trying to predict interactions between proteins. Yeah, they use it a lot in antigen detection to select candidates for immunization, for vaccine production, right? But for that, you need to have a lot because we still don't understand exactly how different surfaces of protein interact with each other to produce tight links between them, like immunoglobulin and the antigen, how it happens. We kind of have an idea of what interactions take place, but we don't know exactly how they're going to happen in real life. So what they're doing is that since they don't know exactly and they don't have a model, they just use a lot of information available online or from high throughput experiments saying, all right, all these molecules were aligned to all these molecules, and we saw this strength of attachment. So let's just let the artificial intelligence make up its mind of how it works. It doesn't matter to me how it works, and let's see if the prediction is high enough. That's basically what they're doing. Yeah. So all these alignments, etc., they serve a purpose to find conserved molecules. And as David was explaining yesterday, there are many ways to use them. Yeah. One of the ways is to design primers, degenerate primers, to tackle several sequences at the same time. We just look for the most conserved regions in these proteins, and we design primers from those amino acids. Or we could also use them for phylogenetic inference. Or we could use it for... That's all I can think of right now. Yeah, but if you go look online, you'll probably see like 100 different usages for having multiple sequence alignment. There is a reason why multiple sequence alignment is one of the most used and the first developed tools in the bioinformatics toolkit. Now we're going to change subjects a little bit. We're going to start seeing transcriptome analysis. Yeah, this is for the rest of the day, we're going to try to understand what transcriptome analysis is, what are the caveats, what are the advantages of using it, yeah, what are the limitations, yeah, trying to understand what's the raw sequence going to look like, what do we have in that raw sequence, and how can we use it? How can we use it in our research? How to design a proper experiment, at least from my part in RNA-seq, differential gene expression, which David doesn't trust at all. And David is going to show you how to use the same information in network analysis, which I don't trust. No, just like. Yeah, but these are different ways of treating the same data. Yeah, and we're going to show you how to do it. You're going to do it yourself I've created a small toy set from Arabidopsis that you copied on your INEA 08 directory. I should probably have erased it, but anyway. There we go. I think I can wrap this up before the break, so that if we have any theory that we need to cover before starting the practice, we can just talk about it during the break and a little bit after. So transcriptome analysis. Could anybody here raise your hands if you're planning or have already designed or are planning to design a transcriptome analysis, RNA-seq analysis at all? If you're expecting to use it sometime in the near or middle future? Yeah, 1, 2, 3, 4, 5, yeah? Yeah, so we reckon that this is one of the important steps that we need to cover here, right? What do you understand by transcriptome analysis? What is the transcriptome? Please, if you want to share, please just use this. Don't be afraid. There is no right or wrong. Well, there is right and wrong answer, but I want to understand where we're coming from.
El efecto de la expresión de algunos genes ante un evento, un desafío, un experimento.
Yeah, that's the effect. Yes, that's a good first glimpse. But what is the transcriptome itself?
It's a gene expression profile from a cell in a specific time in a specific condition. All the genes.
Yes, yes. That is closer. Yes. There you go.
Okay, for a specific cell, transcriptome is all of their RNAs. You have to sequence, well, you have, if you want to know, but transcriptome is all of their RNAs.
Yep, yep, that's adding some more information. So basically, yes, the general idea is correct. The transcriptome is the set of all active transcripts in a specific cell or tissue at a specific time under specific environmental conditions. Why is it so important to phrase it like that? Because if you change one of these parameters, the transcriptome landscape, which combines not only the sequences, but also the abundance, the relative abundance of these sequences, yeah, is going to change. All right? If you change the genome, the transcriptome is going to be different because you have a different set of transcripts, you have different conditions, you have different controllers, you have different promoters, etc. If you change the temperature, if you change the atmospheric pressure, if you change the time of the day length, the day length of your plants, if you change the access to water, etc., all this is going to have an effect on the way the transcriptome is expressed. Right? This is the basis of it. Now, transcriptome by itself, only transcriptome of the species, means the complete set of all the transcripts that are available in a genome. We're talking about all the tissues in an organism, in every single case or scenario. We want to know exactly what are all the genes or the regions of the DNA that are being active. And that includes not only messenger RNA, that includes ribosomal RNA, that includes transfer RNA, that includes a small RNA, that includes non-coding RNA. That is your transcriptome. All the transcripts are available. you have different kinds of transcripts. Now, what are the objectives for today? The objectives for today is going to be, first, that we understand the principles of RNA-seq. What are we getting? Why are we getting that? Yeah, how are we enriching or not enriching our libraries? Yeah, so that you know what is the data set you will be working with. Second, identify the limitations of your RNA-seq experiments. What you can do with them and what you can't. How every single step of the RNA-seq method will affect the final output. I hope you will be able to design RNA-seq experiments and perform basic RNA-seq analysis and differential gene expression analysis, even if in the end you don't trust it. All right? So what are we going to touch today? First, we're going to talk about some very basic things. If you don't know this, don't worry, I'm going to go through I'm planning to go through this quite fast, but if you don't know this, if you're not familiar, if there is something not clear, please do not hesitate to interrupt me at any point. All right? So we're going to talk about central dogma of molecular biology, the difference between transcription and translation, the difference between eukaryotes and prokaryotes, what's the structure of the transcripts, and how these structures are represented in annotation formats. Now, how sequencing transcripts have evolved from targeting just one to targeting everything that is available, advantages for RNA-seq, habits of RNA-seq. Now, what's the central dogma of molecular biology? Anybody want to share? I don't know if you all have taken a molecular biology course, but it's just the main idea that there is an order to how things happen inside every cell. The DNA is the map. It tells you what it contains. The DNA has the set of rules, the models of what the genes should be and what they shouldn't, and how they should be treated, how they should interact with each other, how they should be assembled. So from the DNA, we can get a model of what the gene is going to be by transforming it in RNA. The DNA can duplicate itself, which happens during DNA duplication, and it's important to reproduce unicellularly. The DNA can use RNA polymerases to generate RNA from this DNA model, to generate the different kind of RNAs that we know. We can get messenger RNA, transfer RNA, ribosomal RNA. We can get interference RNA, well, a small RNA. We can get non-coding RNA. And there are a lot more RNA molecules depending on who is doing the classification. And we also know that in some organisms, the RNA can be converted back into DNA. For example, retroviruses. Now, the RNA, not all of them, but the messenger RNA will go out of the nucleus, will go through some stages, post-processing stages, and then it will reach the ribosomes. Once it reaches the ribosomes are the actual machinery who's going to assemble proteins based on the sequence that is set on the RNA, and that comes originally from the DNA. These proteins will have this linear layout at first, and as they are being produced, they start folding into secondary, tertiary, and sometimes quaternary structures, right? That three-dimensional conformation that they have in the end, tertiary and quaternary, are the ones that determine how it's going to work. Now, some RNA can replicate itself, which is something that has just been discovered not too long ago. And you have both from the sense to the antisense and from the antisense to the sense. This is RNA-dependent RNA polymerases. Some RNAs can even remove their own codons without the use of any enzymatic machinery. So some RNAs can actually work as enzymes themselves. But the main idea here is that the DNA will give you the model for producing RNA, and the RNA will transfer that knowledge to the ribosome so that proteins can be built. And the proteins are the ones who actually execute things, who actually act on things. Is there any question here? Okay, cool. Now, transcription and translation. Both are complex processes, particularly in eukaryote cells. Transcription. Yeah, transcription, basically, it's just the step from getting a DNA sequence and using the interactions, the strong interactions between G and C, T and A, to actually build the exact same chain, yeah, with RNA. It will go through, I don't know why I didn't put, it will go through several stages. In eukaryotes, you'll usually have just one gene per, one transcript per gene. All right? You will not have multiple proteins coming from one single gene. That's opposite to prokaryotes. Prokaryotes will have multiple proteins coming from the same messenger RNA. Yeah, and also these RNA sequences have many modifications that allow it to survive outside of the nucleus. Because in the nucleus, the cell constantly has surveillance proteins that will try to destroy foreign RNA. And if not constructed properly, your own RNA is going to be target to these proteins, these RNases. All right? So we have 7-methyl guanosine at the beginning, which is a protection in the 5' end. Then we'll have certain consensus or pseudo-consensus regions in the RNA, yeah, which are the ones that are going to interact with the ribosome. These are the ones that are going to help put the ribosome in the right location to start building the protein. Yeah, it's gonna be the one that will give certain stability to the 5' end of the RNA, so that it prevents it from being attacked. Yeah, and the same thing goes to the other end, the 3' end. The 3' end is gonna have polyadenylation. So a lot of adenines are being added to that end of the RNA, so that when they start being attacked by nucleases, yeah, you have enough to spare. And then also another thing that happens is the decision of introns. Eukaryotic transcripts will have introns and exons. The exons will contain the coding sequences, and the introns will not. The introns are going to be, well, more often than not, are going to be involved in some of the more finer, step of transcript regulation. Yeah, but they are not going to be part of the coding sequence. That means they are not going to be part of the final protein sequence. So all these positions, the introns, need to be removed from the original RNA RNA sequence. Once we have the coding sequence, some of them will have antisense RNA, which will let it combine with the sense RNA in the transfer RNA, so that we know that for each triplet, there's going to be a specific interaction that will allow an amino acid to be added to the protein. The difference between transcription and translation is transcription converts or uses DNA to produce messenger RNA, yeah, and then follows all the steps to get a clean, stable RNA that can travel outside of the nucleus. And translation uses that stable messenger RNA to produce proteins, based only on the coding sequence that is in the middle. There are a lot of other things around a transcript, around the messenger RNA, a finished messenger RNA, yeah, that have been either added or modified during these processing steps. Any questions so far? Is the difference between transcription and translation clear? So, transcription, what is going to produce? What's the final output of transcription? RNA. And what's the final output of translation? Proteins. Cool. Now, in translation, after the protein has been built, you also have a lot of modifications that happen to the proteins. Acetylation, methylation, phosphorylation, the addition of sugars. So there are a lot of modifications that happen to the protein. And all those modifications are directed by the protein sequence itself. The protein sequence has conserved signals that tell, hey, I need to be phosphorylated, I need to be adenylated, I need to add this kind of sugar to my sequence at this position, etc. So all these are coded within the protein itself. And they will affect how the protein behaves as well. Now, prokaryotic versus eukaryotic messenger RNA. This is part of what I was telling right just a second ago. It's like prokaryotes will have some modifications to their messenger RNA, but they're essentially converted into proteins as fast as it happens. Why? Because there is no nucleus. Since there is no nucleus, they are directly exposed to the nucleuses that are present in the cytoplasm of the cell, and therefore they don't have the time to go through all these processing steps. Yeah, they have to be translated right away. Right? Another way how they save time is by coding several proteins into one single messenger RNA. This is called polycistronic genes. These polycistronic genes will code for several proteins in the same messenger RNA. Compared to eukaryotes, eukaryotes just code for one single protein in one messenger RNA. As far as I know, unless that's changed, I don't think so. But if there are polycistronic genes in eukaryotes, it's very rare. Now, the only thing we can compare it with is the fact that we have exons and introns. Yeah, exons being the coding sequences of the eukaryote messenger RNA. Any questions? Cool. Now, how do we represent all of this once we've determined where in the genome we have found a gene? We use annotation formats. And the annotation formats, the one that you must be more used to are NCBI or gene bank annotation formats. When you go to a genome and you see, let me see if I can show you an example. You'll see that there are very important parts or important characteristics that need to be recorded about where in the genome something happens. Just 1 second. There we go. So this is probably the most common one that you'd say. If you ever look for a genome or for a sequence, you probably have run into this format, the gene bank format. It tells you everything you need to know, or it's very detailed. Where was it published? When was it uploaded? How was it produced? What is the related method used, etc. And then you got what they call features. Yeah, and they tell you what's the source, and they tell you also. It goes from 1 to 100, 555, 286, which is the number of nucleotides in total. That's the source. That's where all the features are going to be annotated. And it tells you what's the organism, what's the organelle, what's the molecule type, the cultivar where it was sequenced, database of X reference. This is the taxon. Then you have the first gene. And it gives you exactly what you would expect. The gene. It's built from positions 99,091 to 99,894 and joined from positions 71,485 to 71,598. It tells you the name of the gene, the locus tag, if it's splicing or not. This is a note. I'm not exactly sure what the note means in this case. However, it must be a keyword for something. And this is the reference gene ID. Just the fact that it's joining two pieces. Here, it's telling you about the orientation, if it's going into the plus strand or the minus strand. From the look of it, it looks like it's from the minus strand. It's going from the 99,000 and finishing on the 71,000. Oh, it means which strand of the DNA is being used to produce this gene. On which strand you find it. You have the plus strand or the minus strand. Remember that, yes. I'm guessing in this case. Because usually these things here, yeah, from this position to that position, is always positive. Should always be positive. It goes from the minor position to the further downstream position, right? That's the default. But the fact that it's joining it with something that it's downstream, because the 99,000 it's upstream and 71,000 is downstream, it's telling me that it's joining this thing first, and then the downstream part second. It's telling me, oh, it's going actually in that direction. Then we have the CDSs. It tells me what are the sequences of this gene that are actually coding sequences. Yeah, and it's again the same. And you'll see that this time it's just from 3 different regions. So within that gene, yeah, the coding sequences are actually 3. The gene is composed of 2 places, and the coding sequences is actually from 3 different places within those original 2 places. Yeah, the name of the gene keeps the same. The codon starts in 1. The translation table, this is also something important. Not every organism will use the same translation table. We're used to the translation table for us, right? Come on. There you go. We're used to that translation table, where each codon has a definitive meaning, but it's not the same for other organisms. Organelles use a different translation table, just as some protozoarians, they also use them, unicellular free living organisms. Some archaea, they use different protein translation tables. So the individuals that are more farther apart evolutionarily will use different coding tables. That means that a codon that, for us, is translated into leucine doesn't necessarily will translate into leucine in a different organism. Is this clear? Okay. Let's go back to this. Yes. Now, is it transplicing? Yes, because it needs to splice to actually produce the final outcome. What is the product? The product is a ribosomal protein S12. It's one of the proteins that forms part of the complex of the ribosome. Protein ID. If you want to see the protein sequence, you can go to this ID and see all the properties that have already been recorded for this protein. And now in this database, this is the gene ID. And if it was to be translated, this is the actual translation of this gene. This is what after transcription and translation, this is what you get. Of course, some of these things can be guessed or can be recalculated from just the simple indication of where to select the sequences from in the genome, how to concatenate them, yeah, and we should be able to reconstruct this protein sequence. And there are programs who already do that. Yeah. So the idea is, how do we simplify the annotation of these genes so that these are human readable? Yeah, they can be easily created and they can be shared with everybody. So everybody can just know what is the annotation, what are all the genes of a specific genome. It comprises several different steps to get in this annotation. Yeah, but after that, you get some different formats. The main format is the one that we're showing here, the GenBank format. It's the most common because it's from GenBank. Everybody started using GenBank before bioinformatics was popular. But now that we need easier ways to access this data, this is not easy to read. You cannot easily teach a computer, hey, read this, and just show it in a graph, or just extract the sequences you want. Yeah, because the format is not stable across different records. Sometimes it will take 6 spaces here before you actually read the keyword, and then 12 spaces here until you reach the first definitions. Yeah, and it's difficult to code. There are some programs that already can read this, but it's not the recommended way to share this information. That's why there are these standard conventions to annotate the genome. Yeah, the standard conventions, it needs to store all essential information, basic essential information that lets you reconstruct all these features and subfeatures in the genome. Namely, genes, transfer RNA, ribosomal RNA, what are the proteins that can be deduced from that, also non-coding RNAs, and just give them proper names. Also, if you have subfeatures of a gene, for example, a gene has a transcript, a gene has a promoter, a gene has a termination codon signal, a gene has codons and introns, or in the case of bacteria, a gene has different coding sequences because these are different proteins. All these need to be recorded in this annotation format. It needs to be flexible enough to also store non-essential information, like namings. Yeah, this gene is called XXX in the database 1, and it's called XXY in the database 2, and the protein is the protein product is called YY1 in the database 3. Yeah, all these different non-essential information should be able to be stored in the sequence in an annotation format. There are 3 main formats. From these 3, we're going to just be looking at GFF. In the practice, you were going to see how we can transform GFF format into GTF format, and then I'm also going to quickly describe the BET format because it's the simplest one. The BET format, it just requires 3, well, worst case scenario, 4 columns, but it could have up to 11. The essential is to have 3. The first one is the name of the sequence where the annotation is going to happen. If you have, for example, after you finish an assembly, you call each of your sequences sec1, sec2, sec3, the name of the sequence you're going to annotate is going to be called sec1. So the first column is going to contain sec1. The second column is going to store where the feature starts. If the feature that you found or that you want to record starts on position 7, then that's the number that arrives there. It starts in position 7. If the feature finishes, it starts in position 11, then the second column should say position 11. And the third column, basic for BET, for the BET format, is where the feature ends. So So as you see, the bed format is just a coordinate system. It tells me the sequence I'm annotating, the start and the end of the feature. That's it. After that, the next optional columns are giving it a name, setting up the strand, setting up a definition, if it's an exon, a CDS, yeah, the feature type, yeah, all these things can be added, but in the bed format, which is the simplest one of them all, yeah, it's not necessary. Then we have the GFF. This is what we call the general feature format, and it's the most flexible of them all because it allows to pretty much annotate everything you want. We're going to see an example on the next slide of what a GFF format has, but it has 8 obligatory columns and 1 optional column. The 8 obligatory columns are in a specific order. If you change the order of that, then it no longer is a GFF file. Yeah, just like in bed, the first one is the source sequence, the name of the sequence that is being annotated. In your example, it was, for example, after the assembly we got the last time, it was node 1 underscore length something underscore coverage something, etc. That was the name of your sequence. It was long and confusing, but that was the name of the sequence. That would be on the first column. The second column would be the source. How did you get this sequence? You can get it, you can put something like, oh, I wrote it myself because I wanted to. Yeah, that's also valid. But usually you've used a program to determine what your feature is. For example, sometimes it's an homology. You found a protein in the database that aligned to this region of your assembly, and you say, yeah, I want to record this. I want to record this alignment. I would say, okay, what is the program that was used? It was BLAST. I just used BLAST to find this, to highlight this sequence. Yeah. Then what is the type? This is the third column. The type, it would be a BLAST homology. It could be a BLAST P homology. It could be a CDS. It could be a gene. It could be a transcript. There are a defined number of different keywords that can be used here. But GFF is flexible enough to accept things that are not keywords. The fourth column, it will be the start of the feature. As you can see, this is the coordinate. Yep, where the feature starts and when the feature ends. This is always, in every single case, it has to go upstream. That is, the start is going to be the lowest coordinate of the feature. The end is going to be the highest coordinate of the feature. It doesn't matter if it's on the plus or in the minus, because that's going to be specified in a different column. I'm going to go into more details in the next one. All those formats are tab separated. That means between one column and the next column, there's going to be a tab. You can open it in Excel. It's not recommended because they can be very large, but you can open it in Excel and you can see it nicely ordered. The GFF is the general feature format. The GDF is the gene transfer format. And the BED is, I don't remember what it stands for. So this is an example of a GFF file. Usually the GFF3 file or the GFF file don't have this header. I've added it just to make it easy for us to understand. Yeah, but if you wanted to add it into your GFF file, you can add it as a comment. Just put a hashtag at the beginning of a line, and it's already a comment. So the first column, seg name, as we were saying, what's the sequence to be annotated? The second column, the source, what program you use to annotate this sequence or to find this sequence, to this feature. The third one is what kind of feature it is: gene, messenger RNA, transcript, 5' UTR, the start codon, end codon, transcription start site, termination codon, 3' UTR, BLAST homology search. Pretty much, there are some keywords like the ones I'm showing here, yeah, but pretty much accepts everything. The GFF3 accepts everything you want to throw it. Yeah, but also it probably has keywords for pretty much everything you want to throw it. Then the start, what's the start position of the feature? What's the end position of the feature? What's the score? If you use a program, it's likely going to give you a score, how reliable that feature notation is. Particularly if you're predicting genes. Yeah, in what strand it is placed. Sorry. Oh, come on. There we go. In what strand it is. And this is important because the start and end are always, as I mentioned, are always going to be from the smallest to the biggest. That doesn't mean that that's the actual direction of the feature. It is just convention. The start has to be the lowest point and the end has to be the highest point. Yeah? But this is the part that defines from what strand I'm getting it from. The frame basically tells us within this feature how many nucleotides do I have to remove from the beginning in order to start coding. So, as you would expect, the frame will only appear in those features that are CDSs or start codons that are going to be translated. Because this is important for translation, what frame you're starting from. And finally, the attributes or comments. This is the flexible part of the GFF as well. The fact that you can add some important information, important keywords, yeah, but it's flexible enough that if they are missing, you won't need them to actually display this property. For example, this feature. For example, the first one is a gene, and the only thing we define is the ID. The ID is gene 1. Why we call it gene 1? Because that's what we came up with. Second line, yeah, it's a messenger RNA. Yeah, so the ID we're calling it G1R1. That's the code we went for, yeah, and then there is another key part, parent. This is a subfeature of another bigger feature. So the bigger feature is gene 1. This is the parent of this one, of this messenger RNA. The one that we defined here. So this, just by the definition of a parent in the comments or attributes, we're saying what's the hierarchy of this feature. This is a child of the gene 1. Then we have transcript, and there's a difference between the messenger RNA and the transcript. The messenger RNA is kind of a theoretical construct. It's kind of like the pre-messenger RNA, the one that hasn't been processed yet. The one that has still introns, codons, it hasn't had any other modification. But the messenger RNA will be the one that has already gone out of the nucleus. A score is usually a measure of confidence. It's if you're using, for example, when you're annotating genes, yeah, you can either use computer models, yeah, and just running on a reference genome or an assembly and say, all right, this model predicted these genes to be present. For example, something as simple in bacteria as ORFs. If you find an ORF, you kind of think, oh yeah, this probably codes for a gene, yeah. But based on the ORF properties, yeah, and the characteristics, the program will tell you, yeah, this is kind of likely a protein, or this is kind of likely not a protein. Yeah, but for eukaryotes, it's a bit more complicated because we don't just ORFs to predict where a gene is going to be. Because ORF will not let you know what's the first codon or what's the first exon. Why? Because the first exon will have the AUG, yeah, the start codon signal, but we will not have the end codon signal because it could have several other exons. The only one that's going to have the end codon or the termination signal is going to be the last exon of your gene. And they could be very spread far apart in the genome. Yep. So when you do prediction of genes in eukaryotes, yeah, you usually rely on external evidence. Not only your model of where exons are meant to start introns are meant to start, what are the places that are likely to be spliced, but also external evidence like RNA-seq, ESTs, protein homology, etc. All these things have to be taken into account to actually give more confidence to your genes. And when you do that, if you have like a lot of evidence showing, hey, yes, this looks like an exon, yeah, because it has alignments to proteins in the database. It also has a start codon. It also has an uninterrupted or reading frame. It has a RNA-seq aligning to it. It has ESTs that support it. You have all kind of evidence to say, yeah, I'm 90% sure it is okay. Yeah, so we can call it an exon. Pretty much every program. Pretty much every program. In this case, I'm just using GeneScan as an example. Yeah, but pretty much every gene prediction program will give you a ranking or will give you a reliability score. This score is going to tell you how reliable this feature is. As you can imagine, each level of prediction will have a different level of accuracy. If we talk about just computational predictions, this is called ab initio gene prediction, which means I don't have any external evidence. I just want to base it on the sequence itself. I want to get the details of the sequence, and from that, I want to guess which are the most likely coding regions, genes, exons, etc. Right? Now, this is very unreliable. In the CDS level, it's kind of okay. It reaches around 70% accuracy, 70. That's the CDSs and exons. In the actual transcript level, yeah, it reaches around 60% accuracy. So you have a 10% drop. Why? Because usually it detects incorrectly which exons belong to the same transcript. Then in the gene level, you usually have around 40 to 50% accuracy. Yeah, because basically it merges wrong exons into the same gene. Sometimes it uses exons that belong to a different gene just because they are close enough. Yeah, and that's why we need external evidence. Because sometimes just because they are close enough in the sequence, yeah, that doesn't mean that they belong to the same gene. It's only external evidence who's going to show you, yes, this is actually a new gene, that it belongs here. Yeah, so when you start assigning all these exons, introns, and subfeatures of the genome that could be accurately predicted into actually larger, more complex structures like messenger RNA, transcripts, and genes, yeah, this is where the problems start. And that's why the accuracy rate for each of the levels in the hierarchy, from the base to the top, yeah, that's where errors start happening and the accuracy starts falling. Yeah, but if you use external evidence, you will have more reliable models. And those more reliable models will allow you to predict the genes in your data set more accurately. Is this clear? Is there anything that you think wasn't clear enough? Oh, frame. Frame means that within this feature, yeah, within the parent feature, for example, the CDS is the child of an exon, yeah, or a start codon is the child of an exon. Yes, within the exon feature, how many nucleotides we need to remove from the beginning to start coding. I know it sounds a bit weird, yeah. Basically, it just means that when you have a sequence, you can start reading from the start, you can start reading from position 1, I mean from the second one, or from the third one. And each of those start points will give you a different protein, completely different protein. Just only one of the, and then you can go also from the other way around. You can start from -1, -2, or -3. Those are the 6 reading frames that you have for every DNA sequence, right? In this case, since we already have the strand, that the strand it's shown there in the 6th column, then you only need to show how many nucleotides you have to ignore in the beginning to reach the coding, the start coding sequence. Right? That's what the frame means. and as it's meant to be, it's only meant to be used for coding sequences, which would be start codons and also CDSs. CDS stands for coding sequence. This is the part of the gene that actually produces or gives you the instructions to build a protein. So, okay. Yeah, we'll quickly wrap this up. So from ESTs to RNA-seq. Yeah, the first explorations of the first ways to explore what the transcript contained was using EST libraries. And we know that if you do a total RNA extraction of a cell or a tissue, yeah, what you'll get is the sequences of what is actually actively transcribed. It doesn't matter what, you just want to have a library that represents what is the most, what is active in our genome. Because the inactive parts at this point, in this point of the history of transcript research, wasn't really that important yet. We didn't understand how important it was. Yeah, so basically we did messenger RNA isolation from, in this case, a lettuce. Yeah, then we did reverse transcriptase to produce cDNA from the RNA that we had extracted. Of course, in the middle step, during the messenger RNA extraction, we had to use DNases because we need to get rid of DNases. We need to use proteases because we need to get rid of possible nucleases that will degrade my RNA. We need to keep it frozen, or we need to keep it at a low temperature to prevent other contaminants or other proteins to actively start degrading what we're extracting. We need to use other chemicals to stop everything that could be happening inside of the cell. We need to use detergents to get rid of lipids that could be attached to the DNA or to the RNA. So all these things need to happen during the RNA extraction. And there are very elaborate protocols on how to do a proper total RNA extraction. Then we need to select what we're going to use from this total RNA extraction. If we want to target small RNAs, if we want to target messenger RNAs, we need to get rid first also of the most abundant class of RNA in any cell, ribosomal RNA. We need to get rid of that ribosomal RNA. How do we get rid of that? We use ribosomal RNA depletion. But the fact is that after all these processes, and in each of these processes we're going to lose some data, we're going to lose some RNA that we were targeting, because these are not perfect molecular biology protocols. They are optimized, but they are never perfect. Yeah? We're losing DNA and RNA material in every single step. Yeah? Then we construct the cDNA library by cloning all those fragments that we have selected, cloning them into bacterial vectors, and then just transforming my bacteria with different vectors each, so that we have a collection of bacteria. Each of those bacteria will have a different fragment inserted in the vector, yeah, and each fragment will represent an active transcript. That's basically what we did in these first two steps. And then the idea of the EST library is using the known vector sequence to sequence the unknown insert. Just like we do with most PCR fragments, well, maybe not most PCR fragments, but when you're trying to sequence something you don't know what it is, and you've cloned it into a vector, it's because you know the sequence of the vector. And if you know the sequence of the vector, you can use primers designed from the vector to amplify the region that has been inserted, regardless of whether you know it or not. And that's the first sequencing libraries obtained from RNA, the ESTs libraries, express sequence tags. You don't. Yeah, this is the first approaches into sequencing RNA. These are just the first libraries that were produced, and you'll see that in NCBI you'll find a specific database that's called ESTs, EST libraries. This is the way they were produced. And as you can imagine, since you can, in the best machines that you find available for Sanger, yeah, you can get up to 32, I guess, 32 sequences at a time. Yeah, so imagine how many different transcripts do you think are active in a human cell at a single time? I think humans have in total around 25,000 transcripts or 25,000 genes, and this is messenger RNAs, not taking into account the small RNAs and everything else. 25,000 genes, and from those 25,000 genes, perhaps around 3,000, between 3,000 to 5,000 are active in any single cell at a specific point, depending on the tissue, depending on the age, depending on the development stage, etc. Yeah, and if you clone that and you sequence it and you do it in batches of 32, yeah, it will take quite a bit of time to actually get the full EST library to be finished sequencing. Now, take into account that an EST library can have inserts of different sizes. Some genes are going to be small, some genes are going to be bigger, some genes are going to be fragmented because of the messenger RNA isolation method that we use, because of the chemicals we use during the isolation. Therefore, most of these EST libraries are going to be either fragmented or the sequence itself, it's not going to overlap even though they come from the same vector. That is because the gene is longer than the actual read length of a Sanger machine. So when we take all this into consideration, yeah, we see that, yeah, the EST is an accurate description. Express sequence tags. It's just sequences that we have detected that are part of transcriptionally active genes. It gives us a landscape, an idea of what is present, but it doesn't actually give us full genes. We could use and we can target some specific clones in my library to actually detect and isolate and amplify and completely sequence a few of those genes, but it's going to be labor intensive, it's going to take time, it's going to take a lot of money. Right? So, for example, once you finish sequencing an EST library, you get your data, which is EST's raw reads, look like DNA sequence, and you use a program, like a very old program like CAP3, which is an assembly program, to try to reconstruct some of the transcripts based on overlappings between different clones. Because we are expecting that most of my RNA will be fragmented, and therefore, even if they come from the same gene, yeah, the different clones will have different fragments of the same gene attached to them. Yeah, and therefore, if I sequence the ends, yeah, I will have overlapping regions. But it's not that simple. The usual output from these CAP assembly programs from ESTs is singletons. Sequences that didn't have much extension to do, because you just couldn't get any overlapping information from that. You would have to go very deep sequencing into these EST libraries to actually get some accurate reconstruction of contiguous sequences. So technology improved. And with technology, by technology I mean sequencers, sequencing approaches. Yeah, and now RNA-seq basically is using the same library construction or RNA extraction methods we already knew, but instead of cloning, subcloning, and sequencing based on the vector sequence, what we're going to do is we're going to use massively parallel or high throughput sequencing machines, like 454 at some point, Illumina nowadays, yeah, to produce millions of reads from all the transcripts or the transcript pieces that are in an RNA extraction. And we do pretty much the same. We do the RNA extraction, we do the cleaning in the lab, we do the reverse transcriptase, yeah, and after we got cDNA, instead of cloning them, we start adding adapters, we start adding primers, we start with the library, with the next generation library construction, depending which machine you're going to use. Now, once we have the total RNA, we have a poly-A enrichment. If you want to target messenger RNA, ribosomal RNA depletion, just to get rid of those the RNA that is the most abundant in any RNA-seq extraction. Then we use RNA fragmentation because most genes are going to be above the threshold of sequencing of a Illumina machine, for example. Yeah. Then we do size selection. Then we do reverse transcription using random hexamer attached to platform-specific sequencing adapters. This is basically just to create fragments of DNA that have the adapter that we need to capture them. And we're using a random hexamer to just amplify from anywhere in the fragments that we're using. Why? Because we've already done a poly-A enrichment and we've already done a ribosomal RNA depletion. So we've highly selected messenger RNA in this case. After that, we just clean strand switching using the GGG attached to the second platform-specific sequencing adapter. This is just a trick in molecular biology that we use just to select the plus strand and be sure that we are only sequencing one of the strands of the messenger RNA. Then we do the size selection. And finally, this is ready to go into the sequencing machine. So what we have is in every single step of this library preparation protocol, we are introducing biases. We are introducing potential errors. And we are losing some information that just cannot be captured by the method in the lab we're using. So these are all the things that we need to keep in mind, because this will have a final effect in what conclusions we can draw from our experiments. Any questions so far? Yep. Yeah. We're going to see that in the next slide. Or just come to that in the next slide. Yeah, but yes, she's right. It's like every single organ in the plant will have a different expression profile. It's not going to be the same profile if you sequence the leaf, if you sequence the root, if in the root you sequence a node, yeah, or the actual tip of the root, or the base of the root, if you sequence the stem, it's everything going to be different. It's the same. If you, from a leaf, you select the meristem, or you select the, what's the name of, I just missed the name. What's the name of the ones that allow gases to go in and out? Yeah, those are one. They're going to have a different expression pattern. Everything is going to have a different expression pattern, depending on what the function is. Yeah, and that's what makes it so difficult, yeah, to try to just isolate one set of genes, yeah, from just a DNA extraction. Because in the end, what you got, once you got your tissue and you do the extraction, you build your RNA-seq, basically what you have is a description of a collection of different tissues. Even if you're the best surgeon in the world and you manage to cut just the one organ that you wanted, yeah, you will always have some small number of cells coming from different tissues. You cannot just split them apart. Yeah, you have to take it. Yes, this is part of the error that is going to be attached to my RNA-seq data. The fact that I try to isolate this tissue as much as possible, yeah, but there is always a limit, a physical limit. Nowadays, we were talking a couple of days ago with some of you, yeah, that right now there is the single-cell RNA-seq experiments, yeah, where RNA extracted from one single cell can be tagged and follow through the sequencing process until once you get the sequence, you know which sequences belong to which cell, or to one cell only. So that way you kind of, the methods still need to be developed, but you would be kind of able to differentiate different cells from different tissues based on their expression patterns. Yeah, but this is still a couple of years away. The models are still being built, the analysis pipelines are still being tested. Yeah, but single-cell sequencing, it's probably going to be booming. It's, if I'm not wrong, the current conferences, international conferences, are starting to show more and more experiments and analysis pipelines and predictions based on single-cell sequencing methodologies. You can compete? Repeat single cell? Well, that's one of the most difficult parts here at this point. Yeah, right now, single cell experiments, the main drawback is repeatability. But they are working on that because this is part of the advancement of technology. They just need to keep testing different things, different approaches to try to reduce the error as much, try to increase the reproducibility as much, and try to improve the analysis methodology as much as possible.
Yeah, so there is a statistical way to get around what you just mentioned, that if you have different cells from the same tissue. So first of all, you need to know what your biological question is. How do you relate this? What are you looking for? Are you looking at a specific cell, or you're looking at in total at the tissue? And then when you use correlation analysis, you also use the same tissue. And then you correlate your gene expression profiles, and we will see how we do it later on, to your phenotypic traits. And by that, you omit the bias.
So right now, what we've got after preparing RNA-seq, it's small fragments of around 100 to 150 base pairs, yeah, of sequences that belong to all the active transcripts in the tissues that we extracted. Taking into account that we've already removed ribosomal RNA as much as we could in the lab, and we've already enriched for polyadenylated RNA, which is expected to be messenger RNA. We'll get to that in the analysis later. There are some non-coding RNA that has exactly the same properties as messenger RNA, but they don't produce any proteins. Identifying them and separating them from actual protein coding RNA sequences in your RNA-seq data, it's a challenge on its own. Yeah, but at the moment we're just dealing with RNA-seq. So, what are the advantages of RNA-seq? Just think about it for a second. You've extracted RNA from as pure a tissue as you could get. Yeah, you've extracted total RNA, you've selected only polyadenylated RNA, you've reverse transcribed it, you've sequenced it, and you've sequenced it a lot. You've sequenced it so much that you expect that even the lowest transcribed, or the gene that has the lowest transcription rate in your cell has been detected. Let's assume that, right? What do you actually have? Do you have genes, or do you have fragments of transcripts? Do we have full transcripts or just fragments of transcripts? What is the average length of a transcript? For example, I'll just give you an example. In some genes, some human genes can be hundreds of thousand bases long. But the actual transcript, yeah, maybe 2, 3, 4,000 bases long. It's because the introns, at least in the human genome, can be huge. They can be huge, and the same happens in plants. Yeah, so just because you have a big gene doesn't mean you have a big transcript. And just because you have a small transcript doesn't mean that a full transcript that can be sequenced by one of our current sequencing technologies. Except for one. But Illumina, RNA-seq by itself, yeah, this is not going to produce reads long enough to sequence a full-length transcript. You will sequence snippets of it. What's more, for each transcript, you will have a different number of transcripts in your original RNA extraction. You're not going to have the same number of transcripts for ATPase than you have for ribulose 5-phosphatase. You don't. Yeah, you have different number of copies or of copies of that messenger RNA that are active at the time of your extraction. And just from pure logic, the more copies you have in your RNA-seq sample, the more reads you will get in your library, because you'll have more templates available. Also, the longer the transcripts are, you'll have more space to produce more reads. So the longer the transcripts are, you'll also have more reads coming from that transcript. And now this is what we're talking about in the library part. So let's talk about the advantages. The advantages. First, it's high throughput. You have millions of reads that are produced, so you have a good chance, yeah, of having at least one sample of every single transcript that is available in the tissue. Second, it's unbiased. This is a fun word, because it was only after Illumina machines or older machines are actually used in research that you find the biases inherent to each of these machines. There are GC biases, areas that are AT rich that cannot be, or GC rich that cannot be sequenced easily, and that they are not represented as deeply as non-GC rich regions. The same thing happens with the genes. And so this unbiased is quote unquote, because it kind of gives you a representation of all active genes, but there are parts of the genes that are not going to be really represented because of their GC composition. It retains information of the transcriptional landscape, even though there is a bias towards longer genes, yeah, I know, towards longer genes and more active genes, yeah, there is still a direct correlation between the number of reads and the transcriptional activity. It is not a linear relationship, yeah, but there is a direct relationship. And yeah, that's the last part. You have a direct relationship between the number of reads and the number of transcripts for each of them. What are the disadvantages? There are a lot more disadvantages, or caveats in RNA-seq. First, impossible to use pure cultures so far. Technology is improving. We may be able to do it someday. But currently, yes, it is impossible to use pure tissues. Second, it actually measures cumulative transcription of several cells. Since we said that we are not using just pure tissues, these several cells mostly come from one tissue, which is the one we wanted to isolate, but will probably also contain cells from other tissues. There are biases towards more abundant transcripts. There are biases towards longer transcripts. They depend on the developmental stage and the organ that we're challenging against. It depends on environmental conditions. It depends on epigenetic background of each genome.
Why is that a disadvantage?
Sorry?
Why do you put it as a disadvantage?
That's a good question. Not as a disadvantage, but like limitation, things you need to be aware of.
If this is exactly what you want to measure,
Yeah. But you have to isolate it from all other things. That's what I meant by disadvantage in this case, is that all these things, you need to somehow control them in order to make sure that what you're measuring in relationship between your treatment and what you get is real. That the relationship between the treatment that you're looking for and that you're testing and the genes that you have selected after your differential gene expression is real. And it's not the effect of some of these other developmental stage, environmental conditions, epigenetic background. If you haven't been able to control all that, yeah, there is still the chance that those factors are affecting your transcription landscape, and therefore your results are not as conclusive as you expect them to be. And second, and finally, yeah, although the methods for RNA extraction and library construction have improved a lot, yeah, you still have a lot of background noise that we call when we start sequencing RNA. That is, when we start counting how many reads from the same tissue in the same stage, from the same genome, map to a specific gene, yeah, we have a certain error associated to each of the repeats that we have. And that's gonna inform how the data is spread and how we should tackle it statistically. So the more repeats you have, the more reliable the results will be. Of course, you're not going to have 100 repeats of each treatment. That's just crazy. That's just unreal. Yeah, but the current recommendations is to do at least 3 repeats per treatment. You can get away with using less. You can get away with it, but you have to be cautious about the statistics that you're using, and also how reliable your results will be. And finally, the fact that most index journals will not accept your results if you don't have enough repeats to validate your results. Not anymore, yeah. They probably used to. So yeah, we're going to have a break right now. We're going to continue and just play around with the toy set that I've given to you in your INEA 08 file. So if you have any questions before we go to the break. So either everything was clear or everything was confusing. Biases? Yeah, think about it this way. The longer your transcript, the more fragments you generate from it. Think about it this way. The longer your transcript is, the more fragments you can get from it. Therefore, a longer transcript will have more templates from which to amplify before sequencing. And that's the bias I'm talking about. It's the fact that it's longer, it actually generates a bias in library construction that says, all right, just because it's longer, you're going to get more reads. And the number of reads increases exponentially. It doesn't increase linearly. So that's why I'm saying it's bias. Yeah, your statistical analysis does. Have you heard the terms FPKM, RPKM, etc., which are normalization methods for RNA-seq read counts? Yeah, these are basically methods that have been developed to normalize the data of RNA-seq based on 2 parameters. First is the library size, is how many reads have been produced in this specific library, and second, by gene length. It's how many bases or how long each of the genes is. Of course, for this kind of normalization, you need to know beforehand what's the length of each of the genes that you have sequenced, which sometimes you do, sometimes you don't. Yeah, but de novo assembly is one of the applications from producing an RNA-seq library. De novo assembly, yeah, it's an application. It's one of the paths you can follow after you get your RNA-seq library. It's not the only one. For example, as David will teach you, you can go and do a network analysis from it. I can teach you how to do a differential gene expression analysis from it, or we can do just a de novo transcriptome assembly from your RNA-seq. Yes, we can do all three. We can even do a combination of the three. You don't have to settle for just one path. You can do all of them. Yeah, but it all comes down to what are your objectives.